Morning, everyone. Welcome to Forest Genetics 2023. My name's Nicholas Ukrainetz. I'm the president of the Western Forest Genetics Association, and we are co-hosting this event today with the Canadian Forest Genetics Association. This is the third event that we've held together in as many years. This face-to-face conference was supposed to happen in 2021, but of course the uncertainty of COVID caused us to cancel and delay till now. But since then, we organized a very successful virtual student and postdoc symposium in 2021, and we co-hosted a field tour of research trials at the Skamikan Seed Orchard site in 2022. This is the first in-person conference for these organizations since 2019. So welcome, and I hope you enjoy yourselves. We're pleased to host this live conference in Vernon, which is on the traditional territory of the Sylix people of the Okanagan Nation and the epicenter of forest genetics in the interior of British Columbia. That's why we're here. And I joke with people, this conference isn't going to be looked back on for its glitz and its glamour, as you probably already have noticed. It's going to be for the camaraderie, the wonderful banquet we're going to have later today, the after-hour conversations that you have, and some great talks and posters too. So I hope you all enjoy. I'd really like to especially thank Greg O'Neill for taking on the role of organizing the program for this conference. He did an amazing job. So they have an amazing program set up for us today and Thursday, and we have a great field tour set up for you guys tomorrow. So unfortunately, usually this time of year in the Okanagan, you're guaranteed hot, sunny weather. Unfortunately, this week, for whatever reason, you guys brought in some real nasty weather for us. So keep an eye on the forecast and the weather. It's very unsettled, but be prepared for anything. That's what I'd like to say. So we're still going to go out there. We're going to try our best to stay dry and warm, but be prepared for just about anything. Okay, I'm going to hand it over now to my partner here, Brian Barber, the president of the Canadian Forest Genetics Association.
Thank you, Nick, and welcome everyone. There we go. Yeah, thanks for taking the time to travel from far and wide to be here today. We have about 109 delegates registered for the conference, coming from all across North America, Mexico, United Kingdom, Iceland, Sweden, Poland, Taiwan, South Korea, and Australia. So, and maybe a few other places, but thanks for taking your time and effort to attend our Forest Genetics 2023 Discovery and Innovation in Changing Climates, changing climes, and boy, are the climes a-changing. This event would not have been possible without the generous support of our many sponsors, and the sponsor slide should come up now. And I would also, so please give us a big round of applause for thank you to our sponsors. Yeah, it was very, very generous, and this event would not have been possible without all that great support. I'd also like to acknowledge the BC Ministry of Forests, whose logo does not appear, but they've provided significant in-kind support in terms of the planning organization and for hosting the field tours and the lunch tomorrow at Kalamalka Forestry Center. We're here gathered today in Vernon, again, the home of the Okanagan First Nations people, and we were hoping that an elder from the First Nations would be here to provide us an opening welcome, and if and when she does arrive, then we'll just take a break where we're at in the program and welcome Elder Gregory to give us that welcome and blessing for the conference. Vernon is a special place for me, not because work has brought me here for the last 30 years, but family. My mom and her 10 siblings lived and worked on a farm, Ukrainian immigrants, and just downstream from the same Vernon Creek that flows through this hotel. So I feel very, well, I also grew up in Kelowna and learned to hunt and fish in the surrounding hills. So this is a special place for me. So I'm very grateful to be able to live, work, and recreate on the lands of the Sylix people. Yeah. Now, some housekeeping. In the event of an emergency, fire alarm goes off. The exit doors are to the south of the building here. So just go straight through the kitchen foyer and straight out the double doors, and then gather on the far side of the parking lot. We'll muster there, okay? Far side of the lot, get away from the fire trucks. If you require first aid, just shout. There are several certified first aid attendants in the audience, and we'll get you attended to as soon as possible. Washrooms are located across the hall here. There are also washrooms located between the atrium and the hotel desk. Your program, there are some handouts with just titles of the talks and names, and not the locations of the room. I'll get to that in a bit. But we have a very tight schedule, so please pay attention. All the keynote and plenary and concurrent session A will be in this room, and then the concurrent session B will be at the end of the hall in ballroom 3. Okay? Speakers. If you haven't done so already, please provide your presentations to our AV techs, Bryn and Ben and Aaron. Good. And so you can do that during the breaks, lunch. If you're going to be in ballroom B after this morning, then just give it to Bryn in ballroom 3, session B. They'll also set you up with microphones, lapel mics. Moderators, you can use roving mic. And we're also going to have one of our students, running around and for questions. Yeah, so Pierre Alexander's got a mic, so he'll come around. So please use the mic, like speakers and people asking questions, please use the microphones, because we are recording the sessions. And those sessions will be posted at a later date on CFGA's YouTube channel. So anyone can watch it at their own time and at their own pace and of their own interest. So if you have any problems with it, or if you have any concerns, or you wish to have your presentation posted, or a particular slide because you got unpublished data on it, let us know. I think those particular slides, we can probably blur them, so you don't have to worry about that. Okay? So it's consent by, silence is consent unless you tell us otherwise, okay? Yes, also speakers, when you're up here, there's this confidence monitor. So you don't have to crane your neck and look at the screen. So please use the confidence monitor. You will look and feel more confident. And for students too, because we have a number of students presenting both oral and poster presentations today, and there are prizes on offer. So yes, you want to look confident in your presentation. There are several unnamed judges in the audience who will be assessing you on your rapport and your engagement with the audience, not just your technical content you're presenting. So good luck with that, and I wish you all the best. And for others, please engage the students during the conference and support them, make them feel welcome. They are the next generation. Yes, and on the students front, we have 4 students sponsored by the Canadian Forest Genetics Association, and it's Martin and Jesse and Denise and Pierre Alexander, and so we've supported them. They're recent undergraduate students. They've just graduated and they're all segueing into a master's program. So we're happy to have them here, and please make them feel welcome. Yeah. So you will have met them at the registration desk, and you will have the opportunity to pick up a handout with the program, a bag, a water bottle, and these wonderful name tags, which after the conference will serve as a coaster on your desk. And the logo, can you scroll through on the next slide there? Yeah, so the logo was designed by Gayatri Dinesh of Keynote Web Solutions in Victoria, and that's the company I use to manage the Select Seed and the Forest Genetics Council website and do some program, like brochures, etc. So it was a nice design with 3 trees, the crown formed by the double helix of DNA strand. And soon after we posted this, logo, some astute person noted that the spiral was spiraling in the wrong direction, or so it appeared. And so as you know, all DNA spirals clockwise to the right. And so we flipped the image. And so in recognition of that, I'd like to thank Andy Benowitz for pointing that out. Thank you, Andy. Okay. Now for the important stuff, food. Coffee served in the foyer. We will have lunch next door in Ballroom 2. It will be served early today at 11:40, well, depending on when we finish. And if you didn't request a vegetarian or other dietary needs, please leave those for those who did. The banquet will be hosted by the Vernon Seed Orchard Company tonight. It's located, can I have the next slide? There's a little map. That's not mine. Yeah, it's located about 5-10 minute drive away up on Bench Road. The buses will be departing at 6 p.m. today, so don't be late. And then we'll probably have a staggered return after 9. There will be live music there and a fantastic view. Hopefully, we can avoid the thunderstorms that are rolling through, but we will be under cover, so don't worry about that. They have prepared a wonderful evening and food for us. There will be complimentary wine, and then some 1 beer or cider each, and non-alcoholic drinks. If you would like to have more than 1 beer or cider, please bring cash, $5 a pop, because there won't be any credit cards accepted up there. So hit a bank. I'm telling you that now in case you want to hit up a bank machine today. We'll provide a little bit more information at the end of the day and reminders, and then also information about the field trip that's going to happen tomorrow. All right. Last thing on the food, if you would like to fuel up before the field trip tomorrow, because there won't be any coffee service here, is the hotel is offering a buffet breakfast for $19 plus tax. Buy the coupon at the lobby, and so they know how many to expect for breakfast. And you can also purchase breakfast for Thursday as well. So that's, again, $19 plus tax at the front desk. All right. Oh, there's another 5 there. Thank you, Tia. All right, in case you're, if you come on the bus, you don't have to worry about it. If you want to drive up there, there's the map. Yeah. Thanks, Tia. Yes, and the other thing is, if you're not going to attend the banquet, please let me or Tia know this morning, because we're all going to be a little tight up there. But if you're not attending, then we would really appreciate knowing that in advance. People that have purchased a companion ticket, you don't need the ticket, but there is one at the desk for you if you want a receipt. Okay. So unfortunately, we don't have Elder Gregory here at this moment, and we also have regrets from the City of Vernon. We were going to have either the mayor or a councillor attend, but they have passed on their regrets. So you just had Nick and I here to welcome you. So I will then introduce our first keynote speaker. Dr. Jennifer Grenz with the Faculty of Forestry and the Faculty of Lands and Food Systems. And Jennifer's got a very interesting background from the Lillooet First Nation, is that correct? Lytton. And, okay, Lytton, which of course, if you haven't been watching the news or been driving through, devastating fires a few years ago. And so maybe you'll touch on some of that today as well and the impacts. I'm really fascinated. I didn't talk, we had dinner last night and it was great to catch up. But I wanted to share a little interesting story that I'd learned in my travels actually with Jack Woods. We stopped at the Alkali Lake Band because we were looking for seed orchard properties and interested in maybe First Nations opportunities to get involved there. And Gord Chipman, the forester for the band, he took us to one of the pit sites just above the Fraser River, beautiful grasslands. And he said that they had identified the location. Yeah, they identified the locations of some of these settlements by the presence of basket grass. And am I going to steal any thunder from your talk? Okay, I'll leave it there. I will leave it there. So there, what your appetite. So without further ado, Dr. Jennifer Grenz.
If I come near this microphone, it's not going to get angry at me? Okay. Good morning. Thank you so much for inviting me here today. I just want to acknowledge that this morning we are all gathering on the unceded traditional territories of the Sylix people. And I'm very grateful to be a guest on their lands and be able to share with you a little bit about what I'm working on and what I know. A little spoiler for you, I am not a plant geneticist. So you have a way greater depth of knowledge than I do. It is most definitely not my wheelhouse. However, the work that I do alongside a number of indigenous communities in so-called British Columbia now has my lab learning to incorporate these tools to fill some of the knowledge gaps that have happened as a result of colonization, as well as to affirm the knowledges of our elders, our stories, our oral histories, all to aid in the reclamation and revitalization of our stewardship, using the tools of what my very good friend Jared Williams, Quistanakan from Cowichan Tribe, says is the tools of the new and old ancestors. So I like to think of what you are all doing as the tools of the new ancestors, to help us understand our lands better and make decisions. So my hope for all of you today with what I'm presenting is that you all maybe leave here seeing and thinking about the lands that you're working to bring healing to a little bit differently, and that I'm just giving you something to pause about, again, just in how you see and think about the lands that you work on. So to properly introduce myself is incredibly important as an Indigenous person. As my auntie always says, the way Indigenous people introduce ourselves is, where are you from and who's your mom? So I'm going to tell you where I'm from and who's my mom. So my family comes from the overlapping territories of the St'át'imc, Secwepemc, and Inglekatmik nations. So all those kind of misnomers about Indigenous people that we come from one place is really not true. We come from a multitude of places depending on where we gathered, but my family are members of both the Lytton and Bonaparte First Nations, so Inglekatmik and Secwepemc nations, and we were sort of in the Pavilion and Clinton and Lillooet areas mostly, though we do have some relatives that appeared in the 1881 census in Lytton. And as we were sort of touching on earlier, no one ever knew where any of this was, and I was in Georgia presenting recently, and the Lytton fire news actually made it to the state of Georgia, and people actually knew where Lytton was. And I'm going to talk a little bit about some of the work that I'm doing to help my home community now there in wildfire recovery, as well as the Statlium Nation, where many of my cousins are in the same thing. Oh, and there's the confidence monitor, which is really poorly placed, FYI, because you can't see it when you're standing behind the podium. Okay. Who's my mom? Okay, that lady down in the bottom in the center there that my kids who are much younger than are hugging, that's my mom, Wanda Robinson Peterson. This is my grandmother, Hester Swartz, and my great auntie who's still alive today, and the matriarch of our family in the top right, Louise Swartz. In the middle, I have my great grandma and grandpa, who I was very fortunate to know and spent most of my early childhood in her garden, which I blame her for what I do today, and absolutely loved that. So, and then on the top left there is actually my great-great-grandmother, Hattie Davis, who actually ran a hunting lodge in the post-settler era near Clinton, BC. She was like a total badass. So I just wanted to point that out. But that is who I am and where I'm from. What do I do? So I have the Indigenous Ecology Lab in the Faculty of Forestry and Faculty of Land and Food Systems at UBC. Our work is entirely in service to Indigenous communities. And so what that means is all of our work is Indigenous-led, and actually that we are invited into communities to help solve land healing issues. And so we cover a very broad research base in order to respond to those needs. We're situated within the below ground ecosystems group because most of our work is focusing on plant-soil interactions and soil microbial ecology to inform land healing efforts, as well as the impacts of invasive species and understanding what our culturally important plants need to thrive, which many people refer to as native plants. And if you do some digging through the literature for a lot of our culturally important plants, you will find that there is next to nothing because we sort of take for granted native species. So, you know, we are working a lot on seed ecophysiology and again, just understanding, you know, what our plant relations need to thrive. And I need to share that I didn't come at academia in sort of a normal route. I had a career for 20 years as a restoration ecologist, working for various government agencies and nonprofit organizations, and it's literally the best job ever. Why did I leave it? I don't know. No, I do know. I do know. I left it because I was constantly following this restoration formula of remove invasive species, plant native species, and apparently we were supposed to be returning to Eden. But no, it kind of felt more like this, and perhaps some of you can relate to that, it was just more of an exercise in chasing our tails. You know, we have reinvasion, secondary invasion, you know, and despite well wishes, watering, and fertilizer, many species just failed to establish. And so I wanted to come back and find out why. And this is sort of my knowledge cycle journey, is, you know, out there in the bush, weed whacking and trying to solve problems on the ground, to now taking what I've learned and helping the next generation maybe do it better and feel like they're making more of a difference than I feel like I made over those couple of decades. And in that work, I've spent a lot of time with knowledge keepers and elders over the last 8 years or so, trying to understand how we see land and how we see water and how that has an impact on the decisions that we make going forward. And what I really found was the impact of stories on how we heal land, including the stories that were brought by settlers, including what I refer to as Eden ecology, where the foundation of modern ecology is sort of resting upon this idea of untouched, pristine wilderness, and that we're trying to get back to some arbitrary point in time, despite the fact that we all know that these are very dynamic systems that have experienced changes in speciation over time. And it really conflicts. The more you more time you spend with knowledge keepers and elders, you realize that this actually really conflicts with indigenous ways of knowing, because adaptation, as one of my sort of primary elders said to me, is adaptation is who we are. And he was pointing out to me that perhaps western red cedar would have been considered an invasive species 5,000 or 6,000 years ago, and that sort of changed my perspective. And so trying to shift away from this notion of, naturalness that kind of arrived with settlers as they hit our shores, in Coast Salish territory, I think about, what their perceptions were. And so you can imagine hitting shores there and thinking, like, this is a vast, untouched wilderness with so many resources. You could really understand that sort of feeling, especially when you're used to thinking about food systems totally differently, right? So yeah, food system looks more like the bottom right there, cleared land, agrarian crops, but what they didn't realize when they hit the shores is that this kind of land was actually really sophisticated food systems and technological systems and medicinal systems. And I actually think it was this kind of split of food systems from how land was perceived by settlers that has kind of got us on the wrong trajectory in spite of all of our good intentions. So it's really important that we step away from the notion of hunter-gatherers. It's a notion that perpetuates today even among some indigenous people. It did even for myself, except that the more I learn, the more I realize that our food and technological systems are just as sophisticated as the agricultural and even forest systems, commercial forest systems of today. And so, things like berry production was not just a matter of going out and finding berries and gathering them. These were really sophisticated gardens. I spend a lot of time in Shimshan territory now in what we call forest gardens, where we see evidence of irrigation in these gardens from springs and trellising of berries. And then there are other examples here, like camas as an example, produced by Cowichan tribes. Nancy Turner's done some incredible work to estimate the number of bulbs that would have been produced. There was an economy of camas. This was a major starch that was produced for the nation and shared even with my nation. Clam gardens, Waparo gardens in Coast Salish territory. The more you learn about what these systems were, the more you realize they are everywhere. And I'll give you an example is like the clam gardens as well as fish traps. So I've been fishing on the coast of BC since I could walk and had never noticed clam gardens before until an elder pointed them out to me. Now they're everywhere. You can't miss them, right? So aquaculture, not new, not brought with settlers, but was in fact done by indigenous people for thousands of years. And our work in the Getsaic forest gardens of Kitsillis First Nation, show us, and LIDAR is like such an amazing tool. We can see where village sites are, and of course, every village site, now we're finding these forest gardens. The species composition is completely different than the periphery forest, as are the soils. And then when we do some floating of, I'm spending a lot of time in the hearths of people from thousands of years ago now to see what they were eating, what they were cooking, what they were processing. And when we're floating seeds and doing paleo-ethnobotanical analyses with folks like Natasha Leon's there, in one float we found 19 plant taxa in a single test, charred hazelnut fragments, Pacific crabapple seeds, different rubus species. So this also gives us an idea of, what plants were being used and by whom. And so something that I never used to do in my restoration ecology days was to honor the stories of the past, to learn about the places where I was doing work. Who was there? What was happening? What plants were there? What were the plants used for? I'm never working alone now. I'm hanging out with archaeologists and anthropologists, people that I was kind of taught to be highly skeptical of, but now realize the importance in filling knowledge gaps, that have been lost over time. And I'm also spending a lot of time with other storytellers, as I call them, but this is an exercise in interdisciplinarity. We're hanging out with historical ecologists, archaeologists, plant knowledge keepers, soil scientists, plant scientists, all on one site just to really understand the story of the past so that we can understand the story of the present and then make some decisions going forward about who are the plant relations that should be there now. What do we want these systems to be going forward? And this also involves spending considerable time with elders and knowledge keepers. So this is probably one of my best friends in the whole wide world, Dr. Arvid Charlie Luschim. He is a plant knowledge holder for Cowichan tribes. He's in his 80s now. He was raised by his great-grandparents, which was actually a very common practice in Cowichan because you didn't want knowledge to be lost, so you would give your firstborn child to the great-grandparents so that the language and knowledges would be passed on. And if you know any history of Vancouver Island, you'll know that Cowichan tribes was the last holdout to be settled. And so because of that, his knowledge spans pre-settlement time. And so he is just such a wealth of knowledge in terms of, again, understanding who our plant relations are, what stewardship techniques were, and especially how language connects to understanding plant genetics. And I'm going to talk a little bit more about that in a second. And so we also are working to fill gaps on, understanding plant relations, plant practices. This is rice root on the left. And in that, too, we're learning about, how plants were moved around and used. And there's often sort of a reference. So you harvest the little bulbets off the plant, and then you stick the plant back in. And there's a number of languages that describe this either as the grandmother or the great or the grandfather, and that you put it back in, and again, it will provide progeny going forward. And then we've got elderberry, but we're just trying to learn more about these plants and then fill in things like this. This is the food cycle of Cowichan tribes, and this was all sort of put together through this kind of work to understand what a seasonal food system looked like, what happened at different times of year, all to just give us context that when we face systems like this is highly degraded Gary Oak system, we can decide what happens next, because conventional stewardship techniques continue to fail us. So you can see here, this is on the right-hand side is snowberry that's encroaching on the Gary Oaks and actually inhibiting sort of the next generation of Gary Oaks from going forward. We can't use fire anymore in this particular area because there's a surrounding neighborhood, so what to do? And an elder said to me, this is a Gary Oak system, a camas farm, they call them the camas farm, that doesn't have any children. The next generation is missing. So what do we do about that? And so far, everything that's been tried is not working. There's a mowing trial that happened, but that wiped out all the young Gary Oaks. We can't manage the snowberry as an invasive species with the invasive species program because it's not an invasive species, it's a native species, right? So we have like regulatory and policy hurdles as well that we're dealing with. So we're working together to try and recreate stories going forward while also confronting, how we evaluate success. And so I'm very sorry if anyone, might be proud of this that's in this room, but this is a conservation area. This is one of the Camas or Gary Oak conservation areas near Duncan, where the bulb health is actually diminishing because though the meadow species have been restored through the hard work of many volunteers, this aesthetic-based notion of success is hugely problematic because camas requires human relationship. It was harvested. And now the bulbs are overcrowded and not doing as well, especially with the extreme heat that's happening. And so the bulbs are becoming kind of shriveled, and you can just see some of the impacts of crowding that's occurring. And so, we want to confront these sort of what the elders call colonial success measures, because now we're starting to see some of the impacts of just, whoa, we want this meadow, without understanding that those meadows were actually farms. That's what they were. And so how do we reimagine human relationship back into those meadows to go forward? And this is just some of the Gary oaks also that were planted that died, and so we want to do better. So how we're doing this is applying what I refer to as an indigenous ecology, which we've defined as relationally guided healing of our lands, waters, and relations, which brings a desired balance to an ecosystem while respecting and honoring our mutual dependence through reciprocity. So how do we action this? Well, we have to understand who these plant relations are, who these tree relations are. And this is something that became very, very apparent in all the time that I spent, especially with loose cheam, where there are words in Halkaminam that are separate, that mean moving a berry plant from one place to another, moving a plant from one place to another, Haktewitz, moving fertilized fish eggs from one stream to another stream, Punam. These were all hints that we were moving things around. And what's very interesting is a lot of these things moved with brides, something maybe we don't want to talk about. But there was a recognition of, and there's a word in Halkaminam that's failing me right now that means you need fresh blood. And that's how it's described by a lot of the knowledge keepers and elders. And so they were moving seed clams, fertilized fish eggs, and plants up and down the island with brides because of the recognition of the importance of genetic diversity. And so the more we talked about that, the more we talked about, well, what specific plants are in specific places? Do we remember that? Are there plants in specific places where we're like, that's weird, right? Maybe we should dig into that a little bit more. And it turned out the answer was yes. So plant translocation was a really important part of indigenous land stewardship and indigenous food systems. And there are 5 lines of evidence that are commonly used, especially by historical ecologists. And this is through ethnographies, ethnohistoric records. Language is huge, oral traditions, phytogeography, and archaeology. And now we're adding genetic tools to this as well. And this is important to recognize because I feel like people kind of romanticize or add this mysticism to us without understanding really how sophisticated we're still here, and that we're really just like back then the same as now. Like, I'm a farmer. If someone has something cool, I want to hear about it, and I want to grow it, right? And it wasn't any different. Back then, is like, oh, well, that's a really interesting berry, or, oh, that salmon berry produces more berries. Well, I would like to have that instead, right? So again, we have to kind of like reframe things to think of ourselves in this context because it's not going to be all that different. So through those lines of evidence, some of the work that Nancy Turner and Chelsea Armstrong over at SFU have done, they have confirmed evidence of transplanting and human-assisted range extension for 73 species of so-called native plants. And these include a range of growth forms, so trees, shrubs, and herbs. And I just want to highlight a few of these, including one last night. I was sitting talking with Sally. And I had no idea that Sally was involved in this, but the Gary oak. So Gary oak is, known as being on Vancouver Island and also into southern Washington. And it's funny how I came to this issue, but in working with Loose Cheam and the other elders at Cowichan Tribes, I came in to do my PhD work there. as an outsider, even though I'm indigenous. But they were very happy to work with me because they explained that we actually, the Inglikatmik people, had very tight trading relations with Cowichan tribes. So especially as it came with goat wool for things like dried clams, as well as whole cedar canoes that were pulled up the Fraser River to us in exchange for goat wool. So I thought, okay, well, that's cool. I'm in because of our trading relationships, which is really incredible. And what a lot of people don't know is, and this is what was explained to me, is that Cowichan tribe's territory went way up the Fraser River all the way to Yale. And a lot of people don't know that. And so I got a call from a friend in BC Parks who was like, it's really weird, Jen, but there's this, population of Garry Oaks up at Yale. It's like weird outlier population. Like, what do you think? And the elders at Cowichan tribes explained to me that those were placed there as a marker of that relationship and the end of the territory. Meanwhile, I had no idea that there was this whole sort of genetic project going on that also basically confirmed what was told in oral history, that that outlier population was, and Sally could probably explain it way better than I can, related to the populations on the island as opposed to the populations on the mainland in southern Washington. So plants were being moved around for various reasons. Another example of this is beaked hazelnut, Corylus species, and these were used for food, technology, textiles, and medicine. Worldwide, there are over 25 species, but the species that were, there are a few different species that we're finding associated with archaeology sites and indigenous forest gardens. In British Columbia, there are 3 distinct populations, including one outlier way up in the northwest, which is in Tsimshian territory that I'm working on. And we're still continuing to work with elders to track, which species are where and what the relationships between nations were to kind of try and find more. And so far, it's actually really interesting, but the distributions are associated with tribal alliances and trade relationships. I'm going to tell you why I'm telling you about all this in a moment, sorry. Another plant that was moved around a lot that also is genetically important is soapberries. So there are many stories from different nations about specific soapberry plants in specific places. So I'm working quite a lot right now in my home territory, and there are words in my language that I'm just learning, so I'm not going to kind of do a bad job at using the language now. But those are the soapberries on the hot hills. Those are the soapberries in the stream bed. Those are the soapberries on the north side of the mountain. These are different. They have different names. And this is consistent across a number of nations that Nancy Turner has done work with as well. Crabapple is another one that has a lot of obvious genetic variability. So up where I'm working with Kitsilus First Nation, in their language, there are 8 different words for crabapple. And we've been spending lots of time with elders looking at these different species or different plant relations as they're described to us. And they are observably different, or they taste different. So there's like, the translations are the sweeter one, the one that lasts longer, the longer one, and these are all in distinct areas. And evidence of moving plants and specific plants to places are within our creation stories that show the establishment or supernatural origins of the rich diversity of root vegetables. The Botany Valley is one such location. I'm running out of time, so I'm not going to go too far. I'm not going to tell you the story, but some of these stories are captured even by James Tate in his early works as well. It's kind of unfortunate. I have to learn a lot about my own culture through James Tate's experience, but he did do a really amazing job recording that information. But again, these talk about how plants were moved from one place to another and what makes them special. So what does all this mean in the context of restoration? And that's the real reason that I'm here. And the reclamation and revitalization of our food systems and indigenous stewardship. Well, when we're looking at many of these systems deciding what to do next, this is happening more and more over the last 5 to 10 years. There's a lot of ecological restoration happening. Native plant nurseries are popping up, right? And this is a good thing, right? We need sources for these plants. But what elders are telling me is this is terrible because we're sourcing plants from Vancouver Island and planting them in Lillooet. And those are not our plant relations. This is what is being told to me over and over again by knowledge keepers. Those are not our plant relations. And we have a responsibility to the plants that are there now to understand who they are so that we don't introduce things that could perhaps make them vulnerable. When we're looking, especially where I'm from in Lytton, and we have soil temperatures that are hitting 55 and 60 degrees Celsius, maybe we should pay attention to who's already there and how we can use them to help, in wildfire recovery as an example. We have to be working with a greater specificity. So there's a little bit of irresponsibility in some of this native plant nursery stuff to just be taking relations from one place to another. And an example of this that I'm working on right now is this is a Cowichan estuary. And I got a call from the lands and research manager, and she's like, Jen, it's a plant emergency. You got to get down here. I live in Parksville. And I was told by my mentor there's no such thing as a plant emergency, but apparently there was. So I get in my car, I head down there. They're like, there's camas in salt-intruded areas in the estuary. We've not noticed this before. We don't know why we haven't noticed this, but the elders are going to come to the site, and we're going to talk about this. And the elders, of course, they're always like, well, duh. We had a seasonal food system, so when the stuff up in the dry area was done, we were farming down here. Oh, okay. Well, for whatever reason, camas is flowering amazingly last year in the estuary. So then we start to look into this a little bit more, and it's right in salt-intruded areas. It's so cool. You can see it in the soil profile, where in the intertidal areas, and the bulbs are just right there, and they're growing quite fine. So we're like, okay, well, what do we want to do about this? And the elders are like, well, remember, this is different camas. And suddenly, there's a different word for it than the camas that's up there, and no one knew this. It's funny how things get kind of triggered, these memories happen. And they said, well, you need to find out who these plant relations are, that when you do restoration work here, that you're not bringing camas from up on the hills down into here. So this has now opened up a whole sort of plant genetics, project to figure out who those relations are, so we don't do something, as the elder said, stupid, and bring stuff from up in the dry, sandy hills of the Garry Oaks down into the salt-intruded estuary. And so this is also having an influence on wildfire recovery. So I'm doing a ton of work on vegetation trajectories and understanding what our plant relations need up in Statlium and Inglekatmik territory after the Lytton fire, which has left our lands like moonscape. There's 30 centimeters of ash at least. Not a lot coming back in the severely burned areas, of which most of the fires are. And trying to answer this what next question? 3 minutes? Okay, I'm almost done. And so this has led us now to talk about the importance of seed collection and keeping plants in place, and working with nations to build capacity to, bring together knowledges and make decisions for the future that honor those plant relations where they are, and to learn from them, to learn more about the specificity of these plant relations in these specific places. So we're doing some work to understand cardinal temperatures for germination, all part of the seed ecophysiology work we're doing to look at the differences in populations from different areas, to try and support and affirm what we're being told from oral histories and knowledges. And that's across a number of different species. And I guess my message here is just that we need to be working with greater specificity. And so as an example, Kitsilus First Nation has their own garden and are doing their own plant propagation, which is being supported by a couple of different labs to, again, honor plants in place. And we've been doing some field trials where we took plants from other locations and planted them, and then took plants from place. And what do you know? The plants from place are have a higher establishment rate and are doing better, to put it simply. So there's something to this. And we're going to continue working to honor these plant relations that have belonged to these nations for a long time, but then also understanding the importance of, perhaps occasional introductions from other locations. And so just a couple of adorable pictures of my children, picking some of these plant relations, getting to know them better. And I guess I just hope that I've left you all with, thinking a little bit differently about our plants and how we can honor them, in our land healing efforts going forward. And learn from them. And the time I was spending at the Plant Center in Georgia, we were talking about the importance of this kind of work to teach us about climate adaptation, as an example. So, Cook's Gem, which means thank you in my language. And that is how to get hold of me. Thank you. Oh, okay, apparently I have time for questions. Are there any questions? Yeah?
Here's that mic. Thank you. Thank you for that great presentation, Jennifer. Do you think that there's an opportunity to capitalize upon some of this refined historical knowledge to respond to climate change, and how might that be done?
So that's something like CJ Sai and I at the University of Georgia Plant Center are talking about in terms of some of the work that she's doing with CRISPR. And I should say it's probably some of the most sophisticated conversations or progressive conversations that I've had about things like assisted migration are occurring within indigenous communities. And that might be a little bit surprising to some. But yeah, and I think, there's a lot of work, even the work that's being done on Arabidopsis, like, there are plants that can teach us things if we pay attention, and I think we just need to go beyond the species level, and it's really quite surprising, like, I've been taken now to a number of sites by elders where, like, sites are completely dry. It is so hot, and yet the soapberries are huge and they're thriving, and that, so yeah, I think we just need to pay a little closer attention, and I think that it's really important that that work is not exploiting or extracting, knowledges or plants, from nations, but to be working alongside nations. There's a vested interest in maintaining some of our medicines. Like we've talked about, whether we have to let go of certain species, like Western red cedars, front and center for Cowichan tribes right now. Like, what are we doing? Like, are we trying to save it? Are we moving on? Are we, these are really hard conversations. And like in Lytton with wildfire recovery, some of the discussions that I'm having with folks there is like, are our plants anymore? So I think it's like two pieces, right? Is like, what can be there next, but then also what can we learn from the plants that are still there to move forward. Yeah. I'm off the hook this easy? All right, well, thank you very much.
Can we go in the center of the stage so we get a picture?
Sure.
Yeah, thank you very much, Jennifer. That was a great presentation. Really opened up her perspective and think about the consequences of our actions, particularly when it comes to the assisted migration of tree species in BC. So let's have these conversations, continue them. On behalf of the program committee and the host, I have a small gift for you. It's a local wood carving and a preserve from Davidson's Orchards just up the hill there. Awesome. So thank you very much, Jennifer, and we'll get a picture of you as well.
Sure.
If we can kill that light or yeah.
All right.
There we go.
Thank you.
Oh yeah, there we go. Thank you, Jennifer.
Thank you so much.
Yeah, Phil, the bag is ripping at that.
Okay.
Yeah, that's a pollen. That's a the bags that we've got the gifts in, they're a little fragile, but they're, if you saw the presentation yesterday, they're one of the controlled, the bags that we, place on the trees for controlled pollination.
That's awesome.
I'll now introduce our moderator for session one, Dr. Trevor Dirksen, research scientist at the Kalamalka Forestry Center, BC Ministry of Forests. Trevor.
Well, now I have nothing to say because Brian introduced me, other than to say that, yes, this session is sponsored by the Alberta Forest Genetics Association, so thank you for that. I'm the guy that's going to be sitting here moderating with the nasty cards reminding you your time is almost up. 20-minute sessions, 17 minutes, a few minutes for questions at the end. So let's jump straight into it. First up is Jason Holliday from Virginia Tech talking to us about the genomic tools in the restoration of American chestnut.
Well, I was a little bit worried that the transition to a very genomic-y talk would be abrupt after Dr. Grenz's wonderful introduction to her field, but I think that there are parallels because this is a species that was deeply important to indigenous folks in a different part of the world, in Eastern North America, that was lost, and that is a restoration puzzle at the moment. And I'm going to tell you mostly about, well, entirely about our work to develop trees that can resist a pathogen that was introduced that decimated this species. Big green arrow next. Sweet. All right. So when I first moved from BC to Virginia, I continued working on Western species, and I wanted to find a local species because it's just easier. And also conifers are kind of pain in the ass from a genomic perspective. And I live in a mixed hardwood ecosystem. And so the thing that I ended up sort of falling into was chestnut, and it's the species that is, culturally important, very important to folks in that part of the world, even though most of them have never seen a forest that is comprised of 30% chestnut trees, which is what the forests were 120 years ago, at least in the core of the range for this species, before this fungal blight, Cryphonectria parasitica, was introduced from probably importation of Asian chestnuts into New York, and then it spread and basically wiped out the species. So it creates these characteristic blight, these characteristic cankers that encircle the cambium and kill the stem. The roots survive, though, which is the one saving grace of this species. A lot of the genetics, the historical genetics, is still out there in the wild because the roots survive and they produce prolific root collar sprouts. And this is just me trying to figure out what to do with my kids during COVID. And we needed more samples from the county that we lived in, so I took them to find some of these root collar sprouts. And we actually sequenced this tree. So what else is here? Yeah. So probably 1 billion trees were lost completely, but there's tens of millions of trees that persist. So what am I doing about the chestnut? We'll get to that. What are other people doing about the chestnut? Well, it's sort of related. The American Chestnut Foundation has this back-cross breeding program that you may have heard of, which involves a interspecific hybridization with Chinese chestnut, followed by repeated back-crossing to hopefully develop a tree that is mostly American chestnut, but that retains the resistance factors that are present in the Chinese chestnut. Okay, so intercross, 3 back-cross generations, and then an intercross generation. Okay, should work well, right? And we should have trees that are mostly 15/16, on average 15/16 American chestnut. You may have heard about Bill Powell's work to insert an oxalate-oxidase transgene into American chestnut to create resistance, which it does, and it works well. Okay, on the left is the oxalate-oxidase tree, and on the right is a full sibling without that didn't inherit the oxalate-oxidase. And it looks like this is going to be approved at least by USDA fairly soon. So their recent environmental impact assessment basically says we think this should be allowed to be deregulated and planted without many restrictions. It's not the end game, but it's close. So what are we doing to help with this? A couple of things that I'll talk about is genomic prediction to accelerate the back-cross breeding program, and developing genomic tools or genomic data sets to allow us to determine how to best conserve the adaptive legacy that's present in those stump sprouts in the wild. So the problem with the back-cross breeding program is this isn't a very high heritability trait. So it's easy to find those trees that are definitely susceptible. You inoculate these B3, F2 generation trees, and the ones that get massive cankers, those are no good, right? Then you're left with maybe 10% that are maybe, and you want to get down to like the 1% that are definitely really good. And so the first round of inoculation is just on individual trees. You call down to 10%. Then you have to do progeny tests to really get a good handle on what the breeding value of these mother trees is. And that takes a long time, and TACF is a small organization, and they don't have the resources to do progeny tests on like 6,000 trees. Okay, so when Jared Westbrook took over as the manager of the breeding program there, he was interested in this as well. And so long story short, over the last 8 years, we've genotyped like 6,000 of their trees, and operationally applied this genomic prediction method to call many trees that are predicted to have low breeding values for blight resistance. So this is just basically the long and the short of what that looks like. So Jared developed this so-called blight resistance index, which incorporates a number of different traits and tree characters that are relevant to success in the forest, potentially, where 100 is mean resistance of the Chinese species and 0 is the mean resistance of the American species. So the thing that you immediately notice about this is that as you increase your American chestnut ancestry on the x-axis, you decrease your blight resistance on the y-axis. Makes sense, right? The problem is that we don't really have any trees that are in the upper right quadrant, right? We want trees that are mostly American chestnut, not because we're Nazis, but because we want these trees to be well adapted to their environment, right? We don't know the consequences of having lots of Chinese alleles in this environment, right? So we get trees that are about 50% of the Chinese level of resistance. It's okay, but not great. They tend to have a higher level of Chinese chestnut ancestry as well, so it's a little bit more to the left. We have some that maybe. We keep those, and then we get rid of the ones in blue mostly. And one of the interesting things that we have is these so-called large surviving Americans. So they're trees that have got the blight. They haven't been killed, sort of toppled, but they're kind of gnarly. And we have progeny of those trees, and we've run this model on there. So the question I often get is there a natural resistance in the wild? And the answer is maybe a little bit, but probably not enough to premise a breeding program on. So those progeny of the large surviving trees have a mean resistance a little bit more than just your average American chestnut. So those top backcross trees have 50% of Chinese level resistance. Why? It's not supposed to be that way. When they started this breeding program, they assumed that there were three factors underlying resistance in the Asian species, and they had some reasons for that, but it turns out not to be true and not even really close to being true. So we would expect it to be able to fix 1 in 64 of these progeny for those 3 factors, but it turns out that resistance is pretty polygenic. So Jared has done a number of iterations of QTL mapping in this population, and depending on which sort of population you're thinking about, these are just different sort of B1 founder populations. So Clapper is a B1 tree, Graves is a B1 tree. and then everything on the bottom here. And these bars are just intervals where we find QTL for blight traits across the 12 chromosomes of the chestnut genome, and they're kind of scattered all over the place, okay? So that's going to be hard to find the trees that have all of these things together, right? And they're to some extent different depending on who the Chinese great-great-grandmother was. And not only are these QTL scattered across the genome, there's other factors that don't sort of pass the threshold for significance at QTL as a QTL that are contributing to resistance. And so we see significant heritability for blight resistance on chromosomes that don't have any QTL. Okay, so lots of QTL and lots of other stuff that we're not accounting for with QTL. And as you saw in that initial slide with the sort of scatter plot, the more resistance we have, the more Chinese ancestry we drag along. And so this is just illustrating the different levels of Chinese ancestry in red and American ancestry in blue across the genome in two populations that are sort of the core of the breeding program, the Clapper and Graves populations, just different B1 great-grandparents. Great-grandmothers. So, and this would be, if you overlaid the QTLs, you would see that the spikes in red are coincident with the QTLs. So a lot of hitchhiking when you select on those Chinese alleles that are contributing to resistance, you're pulling along a lot of ancestry for Chinese chestnut. So that's going to be a challenge going forward. Oh, there's another disease that affects American chestnut that's actually worse than blight that folks often don't think about, which is Phytophthora, which actually predates the blight and may have extirpated the chestnut from parts of the range that we never knew it really existed in, because it tends to be affecting warm areas of the range, so like lower elevation, more in the south, and it thoroughly kills the trees. It doesn't leave anything to sort of resprout. Luckily, Phytophthora resistance seems to be a lot simpler than blight. And so this is just an example of QTLs for one of the populations that seems to have carried resistance to Phytophthora from the Chinese chestnut progenitor. And on chromosome 5, we see a lot of the variants for Phytophthora resistance explained. Okay, so another aspect of our work is to sort of help facilitate development of those transgenic lines so they can be used for actual restoration, right? So a single transgenic tree is not a foundation for restoring a species, right? We're going to need diversity, we're going to need local adaptation. And one of the problems with chestnut, of course, is that there are no seeds in the wild, and so we can't do a common garden, or at least we can't very easily do a common garden. I like things to be fairly easy, and so well, I mean, you can get a few seeds here and there, but for the most part, you can't get them. And so what do we do about that? Well, if you're a hammer, everything looks like a nail, and I like to sequence things, and that's sort of what I do. And so I thought, well, we can do this genotype environment association approach to characterize adaptive diversity in those wild stump sprouts and develop a strategy for how we sample that adaptive diversity. Okay, we can't vegetatively propagate those stump sprouts like through root cuttings or most other means, but we can graft them. And we can graft them, put them in highlight, and get pollen from them. We can use that pollen to cross the transgenics. And so we sampled throughout the range. I don't know if I can go back here. I can. We sampled throughout the range of the historical range, those stump sprouts, and my student Alex Sandercock, this is sort of boiling down 4 years of his work into one slide, identified adaptive loci through making relationships between climate and genotypes across that data set. So that's the top here. And then use climate variables in conjunction with this method called gradient forest to try to map how that multi-locus adaptive diversity is partitioned across the range. And so this sort of just shows the different areas of adaptation. And then if we want to try to circumscribe zones of adaptation to determine how to do ex situ conservation of this germplasm, so like how do we decide on a strategy to, we sample X from here and X from there and X from, I thought it was going to be more complicated, but it turns out we've got basically 3 groups, the north, the center, and the south of the range. And we can sort of recapitulate the overall allele frequency gradients across all of these sites if we sort of scatter sample 28 trees from zone 1, 27 from zone 2, and 21 from zone 3. That's a fairly simple method intuition that we use to arrive at those numbers. The south is actually highly diverse and kind of what you would expect if it was like the glacial refuge of this species. And so being more diverse, you need fewer samples in order to capture the diversity. However, the diversity from the south has not really been represented, at least in the breeding program to this point. And so it would probably be beneficial to increase these numbers in the south for a lot of reasons, obviously climate change and so on. The alleles there are potentially going to be quite important. One of the other interesting things we can do with that data set is to try to decide where you put these backcross trees. So the backcross trees have kind of a hodgepodge of ancestry from pollen that was sort of haphazardly gathered from rare wild flowering trees, mostly in the vicinity of Meadowview, Virginia, which is where the orchards for the TACF program are located. And so they went out in the forest, found trees, and got pollen from the wild, and then used those to put on the mother trees for the next generation of backcrossing. So what we can do is use this method called locator, which is a method to sort of try to predict where a sample of unknown origin is located in the range of a species. So we train on a set of samples with known origin, this model, and then we can throw new samples at it and say, where is this thing from? So if we do that for wild trees, so we train it on wild trees, and then we have another set of wild trees that were not used in the training, and predict where they go. On the top right here, the arrows are where the tree is from, so the origin of each arrow, they're like paired dots, where the tree is from, and where locator thinks it's from. So there's a little bit of a bias for a locator to want to push them north, but pretty good. On the bottom, the backcross trees, it really wants to push them. It really wants to push them north. So all we know is where the last backcross founder was, and locator wants to push it north, probably because some of the preceding generations, the trees came from further north, like the Graves and Clapper trees are from like Connecticut and Maryland. But it's not clear. Like you have these backcross trees, they have multiple genetic. Multiple parents going back over many generations, and like, what do you do with them? Oh, there we go. I have 3 minutes after conclusions. Look at that. Okay, so there's a lot of other stuff going on in this project. I had 15 minutes, so that's just sort of the long and short of it. There's some hope for restoration of this species. The question of how you do that restoration is not my not in my wheelhouse. There's other folks that are thinking about that. The forest has moved on, but there's a lot of folks who want to see chestnut back in the forest. And so to the extent that we can get trees that are resistant through either the backcross method, the transgenic method, and also through merging those, so by making crosses between the two, we're potentially going to have trees that have some resistance. Hopefully they will be an overstory tree like American chestnut, and not a kind of branchy understory tree that gets shaded out like Chinese chestnut would be if you put it in the forest, the wild type species at least. We need to incorporate adaptive diversity, obviously, and that's going to require more generations of crosses, and just to get the effective population size up. And I sort of already mentioned the last point. So Jared Westbrook and Alexander Cooke were the ones that primarily contributed to the slides that you saw, but a ton of other folks are involved, and lots of different funding sources to try to bring this work to fruition. But we're getting there, but not quite there yet. So thanks.
Jason was right on time. We have a little bit behind in real time, but 2 minutes for questions.
Yeah, just say, note real quick, there is actually a third chestnut program out there that works within American chestnut. I told Dana Nelson about it a little bit. He went out with some people, saw it, and was fairly impressed with some of the resistance too. So I think sometimes we have to be careful how we jump ahead and make certain assumptions. I'm not knocking any of these other two groups, but very often it's dropped that there is also work with American chestnut my facility will host a nationwide resistance workshop in a couple of weeks, and we're going to have a representative from that group there too. So I just want people to know that.
Are you referring to the chestnut cooperators, or are you referring to?
Yes.
Yeah. So this is a program of the large surviving trees where they're trying to make crosses between the large surviving trees, and we're actually starting to collaborate with them. There was some difficulty, let's say, but we're getting to a point where we're starting to work with them. I mean, the royal we, right, like TACF and like by extension, we sequence the trees, and so so we're starting to. Yeah.
That's great. One of the board members will come out to our facility in a couple of weeks and can presentation too. So I just want that to be out there so we have a more holistic view of everything.
I have a question about the ex situ conservation. Of course, a big interest of mine. For a tree that once occurred in the billions, it seems that 20 trees per region is a really low number for sufficiently conserving the genetic diversity. So can you talk about that kind of disconnect, and then also why, even though it's more diverse in the south, you need fewer samples?
Yeah. So one of the things with that is, our goal there was to sort of say, like, what's sort of the minimum threshold that we would need to cross to reasonably approximate what the allele frequencies for these adaptive sites are. And so, it was present in the billions, but it's also highly diverse, highly heterozygous. So it's actually not that hard to capture most of the alleles that are present in the wild with a relatively small number of samples. That's not to say that 20 is going to be the number, but I don't think it needs to be 2,000. And one of the things that's missing in all of this that I talked about was that we're not looking at rare alleles. And so the GEA stuff, if you include stuff that's super rare, then you get lots of spurious results, lots of spurious associations. And so we didn't include them for that reason. But one of the things we're thinking about, I guess I'm supposed to be doing this, but I haven't done it yet, is to try to look at how the different sample numbers that we might choose will affect will kind of truncate the gene trees, and we'll lose those rare alleles. And so what would be a reasonable kind of intersection between, getting all of the adaptive sites polymorphic in our populations, and not losing too many of those rare alleles. And so that's sort of the next step that I need to, when I find some time to sit in front of the computer, I need to think about that as well. Yeah. Thanks for the reminder.
Thanks, Jason. Next up is Janice Cooke from University of Alberta, talking about genetic signatures related to resistance, I think, and lodgepole pine, related to mountain pine beetle.
Jason is always such a hard act to follow. Down would be awesome. And I want to thank Jennifer also for rearranging the equipment. I actually have a mic on. Because, yeah, you're right. It's not the best setup. I'm not a fan of lecterns. I want to thank the organizers so much for giving me the chance to talk about this research day. It's near and dear to my heart. I want to start by acknowledging that we're on the traditional territories of the Sylix First Nation, and I'm an uninvited guest here. I've come from Treaty 6 territory in Alberta, and that's the home of many different First Nations, including the Cree as well as Métis Nations. The research that I'm privileged to tell you about today took place over, and there's more rearrangements, over several different areas across northern BC that are home of diverse First Nations, and we're really lucky that we've had a chance to do that work there. The work that I'm going to talk about today is not all my work. Of course, it took Herculean efforts of a number of different people, and I really wanted to acknowledge my co-authors here, especially Kathy, Rhiannon, and Josh, who did an amazing job of this project and who are all moving on to fabulous positions. And I'm really grateful for their creativity and their perseverance on this, as well as Steph and Dazine and David. I'd also like to point out a shout out to one of my mentors, Patrick Von Etikis. Patrick was my mentor as an undergrad, and my very first meeting that I ever went to was here in Vernon, Western Forest Genetics meeting, and my supervisor said, "You're not going." And Patrick said, "Yeah, you're going, come with us." And it was just as I was moving into forest biology and coming to this meeting, really reaffirmed to me that I had made the right choice to work in forest biology, and I've done so since. And I've turned to Patrick over the years for words of sage wisdom, such as this. This is a quote from Patrick yesterday. And sometimes I've listened to him, and sometimes maybe I haven't, and maybe today I'm glad. They didn't tell me this until after this project actually was well underway. So here we are in Vernon. Vernon is one of those areas that has been extremely affected by mountain pine beetle over the course of the outbreak, which started more than 20 years ago, which is really rather amazing. I'm going to try this. There we go. So this region down in here, here we are down the star, this region down in here, this is a historic region where mountain pine beetle outbreaks are known from the colonialist perspective, went back when we started counting insects and looking at forest health damage. And we also know from oral histories of First Nations people that these are areas that had historically seen these red dead trees. The first part of the present outbreak was really massive, covering much of this historic territory, extending north, and then following a mass in-migration event into the Peace River country, we saw this spread across northern Alberta. And so we're looking at about 20 million hectares of trees that have been affected just in Western Canada. So on the ground, we saw lots of pictures like this in years past. What we saw less of, unless you lived here, were what happens afterwards, where those red needles fall off, the trees are dead. And this happens to be, in Norman Lake Road. That's about 20 kilometers from where I grew up, the house that I grew up in Prince George. And so when you see all of these dead trees in the landscape, you have to wonder, what is the impact of mountain pine beetle on those populations of pine across that enormous landscape? And you know, every so often, you come across trees that are survivors, and they don't show up super well in this slide, but this is one here, and this is one here. So in this mass destruction, these trees that have been killed, there are survivors. And so are those trees just lucky, or is there a genetic component that rendered them to be survivors? So we wanted to get at those questions. And this is where Staffen comes in and his walk in the woods. Ron Sederoff famously said, just take a walk in the woods. We have our genetic diversity there. And look with your eyes and you'll see what you need to see. And so Staffen set out to collect cones from both what we call our victims and our survivors. Not great language if you're trying to get a paper published. But nonetheless, Staffen and his student collected cones from our victims and our survivors and put together a common garden of progeny from these victims and survivors. So we're calling these our F1 MPB killed, F1 MPB survivors. And that gave us genetic material that we could test, because of course, testing dead trees, trying to get the DNA out of them is, you know, probably a fool's errand. But being able to test the progeny, that lets us then apply our genomic tools. So these trees were sampled from our victims and survivors at 4 distinct sites at multiple stands within those sites. So we have 4 locations. We had a total of 45 half-sib families, and we had just about 500 progeny that we ultimately genotyped and passed through all of Rhiannon's strict QA/QC criteria. So these were the trees at the time they were sampled. They were about 4 or 5 years old. These trees are about 11 years old now, and we're actually going to go out this year and do some additional phenotyping. So we wanted to start out with a population genomics approach, and then step into the quantitative genomics approach. And then afterwards, we brought in some landscape genomics, and I'm going to show you why we did that in a minute. So the first thing we wanted to ask is, did mountain pine beetle impact the genetic diversity on the land scale? Was this a natural disturbance of such a force that you actually could see that hand? And it turns out that, in fact, at 3 of the 4 sites, using minor allele frequency analyses, that we do have a significantly reduced genetic diversity. So one way that we can interpret this is that we've got maybe reduced adaptive potential. Another way we can interpret this is in the context of pest-pathogen-plant relations, is as an evolutionary arms race. This idea that these are not separate forces, but one is impacting on the other. So mountain pine beetle comes in, the populations rise to these enormous levels. They're removing a lot of this pine from landscape, but those that survive, maybe they're surviving for a reason. Those are the ones that are living, moving on to provide progeny for the natural reforestation of these areas. Maybe they have some tools at their disposal now that render them more resilient. But of course, those beetles now have many generations that can pass as they're in those endemic phases, attacking the sick, unhealthy trees to become maybe adapted to overcoming those. And so we get this co-evolutionary history, or so the paradigm goes. So one way to look at this is we may have reduced adaptive potential. Another way to look at it is that this is the interrelation between these organisms in this co-evolved relationship. So first thing that Josh did was look at the heritability. The heritability of being a progeny of survivor was 0.48, super high. We were so excited. And then here on the GWAS, and it was kind of disappointing, none of these loci passed our magic line of publication. And so Dave challenged us yesterday to show those results that don't necessarily lead to a pub. Dave, this is for you. But only slightly deflated. Josh went on to learn genomic selection at a workshop offered by Fikrik Isaac and others. And so running genomic selection, now we saw some promising results. We thought, we can't let that GWAS just go. It's too near and dear to our heart. So Josh went on and looked at what the correlation was at a locus level, between the genomic selection and the GWAS. And we see a pretty darn good correlation. And so the genes of largest effect, by and large, in genomic selection are also the genes of largest effect in the GWAS. And so applying some pretty strict criteria, he outlined about 210 of these, what we're calling the genotype by phenotype loci. So then Kathy came in because she loves landscape genomics, and she looked at those loci that had correlation with environmental factors. So we call those our genotype by environment loci. And there were 30 of those, 12 of which were in common with these 210. So this is the kind of math that I can do with at least a pen and paper. You have 198 loci in these loci of largest effect that we're calling predictors of the survivor phenotype. So we're pretty excited by this. So we were moving on now, because this is really, it's just telling us that these trees are not just lucky. There is a genetic fingerprint that separates those trees that survive from those trees that were victims. But we don't know what the phenotype is. And so That's a challenge, right? We've got some genetic differences, but what's driving that? And so we, as I mentioned, we're going back out to the field now that, oops, there we go. I didn't hit the black screen of death, so I'm happy about that. We're going back to characterize these trees, which are now 10, 11 years old, so that's really exciting. But even more exciting was receiving 4,500 seedlings back in March from BC Minister of Forests. A big thanks to Ukrainetz and his team for identifying a number of families from their own breeding programs and their provenance trials where they identified victims and survivors and gathered those seeds, grew those seedlings. We have just finished a very large experiment with those. We're also collecting materials to give another round to this genomic selection GWAS approach to see if we take an independent population, can we again discriminate the victims from the survivors at the level of the genetics, and what traits are different. So once you've assembled a population that's magic like this, people are drawn to it, and so we're really fortunate to have a number of collaborators who are working with us to phenotype these guys to see what is it about host quality. Is it something more obvious like defense? Is it something less obvious like maybe attraction that is leading to a different relationship between mountain pine beetle and the victims and mountain pine beetle and the survivors? So with that, I want to give a big thanks to all of the members of my lab who are participating in the current experiment, and particularly those long-suffering individuals who have been working so long and so hard on this project, and especially to all of our collaborators. Thank you to Sally, thank you to Sam for sharing your adaptree SNP chip. Having community resources like that and community collegiality, super important to make sure that we can make this kind of progress. So thank you.
Thanks, Janice, for putting us back on time. 2 minutes for questions.
Good to go? Okay, thank you so much for the presentation. I know you finished with this question, like what is the phenotype that's going to affect resistance versus susceptibility? But given that mountain pine beetle affects the trees at maturity, do you think that you're going to be able to identify traits in the seedlings, or are you setting up this population for like 50 years down the road?
Yeah, you know, that's an awesome question, because we can't just take mountain pine beetle and willy-nilly attack little seedlings, right? So we can't follow that standard path that most plant pathogen type genetic studies would do, where you do the challenge, right? So we have to get creative. And there's a few ways that we're getting creative. One is using the fungal associate that mountain pine beetle carries, Grossmannia clavigera, as our challenge to these seedlings. So that brings in the defense component. But we know it's imperfect because the fungal pathogen, which is a necrotroph, is inducing a similar response as does an herbivore like mountain pine beetle, but it's not the same, it's not identical. So what we have, actually, and Colleen's going to talk about this because it's a fabulous experiment. We actually, in the bush, we ran an experiment. We compared mature tree responses to natural mountain pine beetle attack versus Grossmannia clavigera inoculation. So we have that as RNA-seq data. That tells us what the commonalities are in those defense responses and what the differences are. We're running RNA-seq on these trees as well. And so we can use that common set that we pull out of the mature tree experiment then to filter the RNA-seq data from these seedlings to say, these are the sets of genes that really interest in as the core response. How are they different between these families? So that's one way that we're doing it. The other way, you know, we often hear that, well, that's a seedling experiment. Our trees, they're all different, right? It's always different, seedling and mature trees. That is true for so many things, but at a core level, there are pretty good commonalities. And now for about 20 years, we have been looking at seedlings in tandem with with mature trees, something that's big enough that mountain pine beetle can attack. And so I have been surprised at how much commonality there is. And in a completely separate project that Laura's going to talk a little bit about in her talk, we have a parallel experiment with Western gall rust, where we've done seedling inoculation, seedling challenges of the same families that are planted out in provenance trials, where they've been naturally infested with Crenartium harknessii. And thanks to Andy for providing his data and the families that we worked with, the correlation between gall formation in the seedlings with this artificial challenge where you're just throwing the hammer at them versus these trees that are being naturally infested over the years, no inoculation whatsoever, that correlation is close to 0.7. So within family, really good. Even between families, pretty darn good. So I think we owe ourselves a bit of kindness on the seedling thing. We can use them if we're creative, and if we think about those experiments really carefully, we can get a lot of information from them that we can't get from that bigger experiment. But you're right. Ultimately, I have to come back in 35 years, and I don't even do the math on how old I'll be on that time, to see how this actually really played out. So for all of the students in the audience, I need to take up this project and use whatever social media we're using. In 35 years, let me know how the experiment is.
Excellent, thank you. Thank you, Trevor. Yeah, Wollongong, that was pretty good. Yeah, today's talk is joint work with Brian, who you'll also hear this afternoon. And I should say that I've prepared this talk, it's algebra-free, so you'll either be relieved or disappointed. So my main background is the analysis of inbred crop data. And so the talk has a bit of that flavour, but I would also like to relate it to your area with trees. So the big difference is in crop breeding, the crops that I've involved in, the aim is selecting the best varieties. And so we run variety trials, and they are properly designed field experiments, and the individual varieties are grown, and they're grown with replication. And so we want to select the best. Whereas in your case, in tree breeding, it's all about selecting the parental trees. So you still have lovely designed field experiments, but the parents are not grown, it's only the progeny. So we're still trying to get at the parental breeding value. So in this talk, I'm going to talk about variety, but you can substitute parental tree or tet or whatever the word is that you might be familiar with. So, well, whatever it is that you're selecting for, the main thing is that you really need to have data from a multi-environment trial. So data combined across a range of trials, and those trials hopefully spanning the target growing environments. In our case, in inbred crops, it's both geographic locations and year of growing season, essentially. So we need to both create a data set that's appropriate for the selection decisions and also have an analysis that's going to do that. And the main reason we need to have all of those trials is because of this ubiquitous variety by environment interaction, genotype by environment interaction, if you like. But the core of it is that the way that some varieties respond differently to a change in environment, and in worst case or best case scenario, the rankings of those varieties change across different environments. And we need to get a grip on that in terms of selection. So the method of analysis that we use in Australia, which has been long established as providing a really good analysis for multi-environment trial data, is a linear mixed model analysis in which we embed a factor analytic model for the variety effects in different environments. And we've used this for many, many years, and we know that it works really, really well in terms of capturing variety by environment interaction. The information that we get out of that analysis, the core information, if you like, is a prediction of the variety effects for every variety in every environment, even if those varieties were not grown in those environments. So we're confronted at the end of the analysis with a massive amount of information. We need to be able to summarise that in some meaningful way in order to make selection decisions. So the presence of variety by environment interaction makes that more complicated, because it's not going to be sensible just to take an average across all the environments, for example. So back in 2018, we developed some methodology which we called overall performance, which unfortunately we use the term OP, which is not open pollinated, but overall performance and stability. And it was all derived from the fact that this factor analytic model that we use, it looks a bit like a multiple regression embedded within our overall model. So we have a whole lot of factor loadings. They are associated with environments, and you can think of them like environmental covariates, except they are estimated from the data. I think someone used the term latent factors. They are actually estimated within the analysis. But let's think of them as environmental covariates. On the flip side of it, we've got the varieties, and the varieties have different slopes. They're called scores, so different slopes for each of those environmental covariates. What often happens in practice when you do this factor analytic model, it's a little bit like a principal component analysis embedded within the model. If all the estimated loadings for the first factor, so the first factor is the one that accounts for the most, it's like a regression, it accounts for the most variation, the second factor the next most, and so on down the line. Often we find that first factor, all the loadings, they all have a positive value. And what that means is that first factor is like an overall average, if you like. Now, I've got yield here because that's the trait that we mostly deal with in our crops. So it might be height, for example, in trees. So that first factor is often all positive, so some sort of general average. And then the higher order factors, which are getting less important as you go down, they're often mixtures, or typically mixtures of positives and negatives. So they're what they're called in the literature as bipolar factors. They represent contrasts between environments, those with the positive versus those with the negative. So in that setup, we used the first factor to define something called overall performance, and stability was all of those all those contrasting factors. That often actually works, like the first factor is often positive, but it raises the standard problem in any sort of factorial experiment. What is the sense in looking at a main effect across all your other factors if there is crossover and interaction? You're essentially averaging over interaction, and how sensible is that? Well, not very, as it turns out. So to address this, we developed this new methodology which forms groups of environments which, and I'm using the words very carefully here, groups of environments that discriminate varieties with different patterns of interaction. Many people have looked at grouping environments and getting estimates of variety effects for those environments, but they often use cluster methods and various things that we might talk about later. The big distinction here is that we're all about the varieties. That's what we're selecting on. They're the most important things, not the environment so much. So what we want to be able to do is pick up varieties that have differential patterns of interaction, and we want to know how they perform. So the actual loadings themselves provide the key to be able to work this out. Once again, we've got, if the factor is all positive, we've got some sort of scale factor going on. And then all of those bipolar factors, they're all about contrasts, positive, negative loadings. What that means is as soon as you've got a factor that has positives and negatives, there are going to be some varieties in that data set that exhibit crossover interaction on that factor, because you will have positives and negative slopes for those varieties. And we've got the standard sort of picture here of a crossover interaction in terms of factor analysis. If we've got, let's say it's the second factor loading, we've got negative loadings on the left, positive on the right. If two varieties A and B have different slopes of their scores, then there's going to be a crossover on that factor. So the rankings are going to change between those two groups of environments. So the concept is as simple as that. We obviously have more than one factor in the analysis, so the way we actually form these groups is by looking at each factor and dividing them up into positives and negatives, and then essentially pasting together the positives and negatives across the factors. So if we've got an FA3 model, for example, then potentially there are 8 groups, but there could be positive, 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 negative, and so on. And by the fact that positive versus negative is where all the contrasts are going on, if we form groups in this way, what it means is we're going to be able to discriminate varieties that have large responses to those contrasts. So, those large scores that are going on. And for that reason, because they're actually picking up variety interaction, we call them interaction classes. Once we've got interaction classes, essentially we're saying varieties are going to perform similarly within a class, but they might perform quite differently across classes. Then it makes sense to have some sort of overall performance within a class, within an I class. Then we can do things like identify the best varieties within each I class, but we could also match varieties in terms of their patterns of interaction across those classes. And the third point is important because what we're doing here is we're not doing a piecemeal analysis of doing a cluster analysis on top of what we're doing. We're actually using the parameters we're getting out of that analysis to form the groups. So an example here, which is taken from a wheat breeding program in Australia. Probably the most important details are we've got 73 environments in this data set, and environment here is a combination of where it was grown and the year that it was grown in. So this is covering a 3-year period. What we're trying to do is select varieties in the late stage. There are 96 of those varieties. This was the example used in the paper that I'll put up in a minute. So to keep it simple, we did not use relatedness between the varieties, but in practice we always do. So it's just keeping it a bit simple. I'm not going to give the details, but we fitted a linear mix model with a factor analytic model for the variety effects in different environments. And we only fitted a model with 4 factors. It accounted for 74% of the overall genetic variance. We would normally push it a bit further than that, but that's enough to illustrate the concepts. And the amount of variance that these 4 environmental covariates, if you like, are explaining are in order about 48%, 16, 6, and 4. So that decreasing order of importance. So what are these? How do we form these I classes? This is what the estimates of the loadings look like. Remember, I had 73 environments. I've just taken a few things here, and if I could figure out what the pointer is, no, there's a danger, let's not worry. We've got our 4 factors in order of importance down there. The first 2 environments up there, everything had positive loadings in the first factor. But the first 2 environments then had negative loadings in the second, negative in the third, and negative in the fourth. So they go into an I class that, we label it as P-triple-N for obvious reasons. The next group down, they're all positive in the first, negative, negative, positive. So it's fairly obvious what we're doing here. And as you can see, that first factor, it was general ability, everything was positive. But the second, third, and fourth, they're all mixtures of positives and negatives with the numbers down the bottom. So we fitted an FA4 model. Potentially there might be 16 I classes, 2 to the power of 4, but only 8, I think it's 8, were present in the data. And these are the number of environments in each of those classes. So they're spread quite evenly in this particular example. What's really important, as I said, we're all about the varieties. We want to select varieties. So information on varieties is the key. So the slopes of the varieties to each of those covariates is really important, the slopes or the scores, say. So if we look at the first 2 factors, on the y-axis I've got the BLUPs for the first factor scores. These are all random effects. On the x-axis, the second factor scores. So because the first factor was general yielding ability, things that are up high are generally good performing varieties. So the few that I've got labelled up the top there are actually, they perform really well. The second factor is contrast between the negatives and positives, and the varieties I've got highlighted there. On the right-hand side, Sting, Scepter and Mace, they all had a positive value for that second score, whereas Ballista has a negative value. So what that translates to is Ballista is actually going to do relatively better in environments that had a negative loading on the second factor, whereas on the other side, the other varieties are going to do the opposite. They're going to perform relatively worse in those environments. So looking at the variety score side is quite important. And you can do that for the other scores. We had 4 factors, so we can go down the line there. I think I'm going to run out of time, so we'll keep going. So the scores are really important, but what they're doing is just giving us information, relative to that factor. When we're actually going to make a selection decision, we need to be able to combine all of that information into one sort of score, one value, if you like. We want to know in tonnes per hectare how these varieties perform. So we can work out overall performance within an I class, which is literally the means of our predicted variety by environment effects taken over the environments in a particular I class. So it's once again a simple mean within an I class. And it's nice to be able to visualise these things in what we call an I class plot, which is literally, our overall performance, tonnes per hectare for these varieties, and the axis on the bottom is I classes, and because of the way they're formed, there's a natural ordering to this. So from the left-hand side to the right-hand side, we're changing from environments that had a negative loading in the second factor to a positive loading. So that's actually where the biggest G by E is happening, from the left to the right, whereas closer together they're only differing in the fourth factor, which is less G by E going on. And you can see all sorts of lovely things here. Probably the most interesting is Ballista, which is the red one, which is up the top there, and Sting is the purple one. And you can see there's that opposite effect. Remember that the scores were different on the second factor, positive versus negative. And you can see here that Ballista does really, really well on the left-hand side of the graph, but it drops down a bit on the right, whereas Sting, for example, the purple, does the opposite. And you also get things like varieties working in parallel. The patterns of G by E are the same. The beautiful story about this is Ballista and Sting. This analysis was done after these varieties were actually released, but way back when the company first looked at it, those two varieties, they thought, oh, they're about similar overall, which is actually the dotted lines, and yes, they are similar overall. They didn't have I classes back then, but they had a gut feeling that these two varieties had a bit of interaction and were forming differently. So they made the brave decision to release both of them, which is an expensive thing to do. And in hindsight now, we've been able to analyse it and actually, back up that yes, this was exactly the right thing to do. Okay. Because we're running a bit of time short, I'm going to keep going, but essentially I want to, reaffirm that this is all about varieties. We're not clustering environments for that sake. We are grouping them to get at variety by environment interaction. So I won't go, you can actually look at correlations and see how they line up. the main thing I want to get out is that we've been using this technique now for quite a long time. A lot of the major breeding companies in Australia are using this technique with pedigree or marker information. Everyone's, they actually really love it because it makes sense. It actually backs up what they feel is going on. So we're quite excited about the way that, it's been able to help in selection decisions. This was the original paper on which this data was presented, but very shortly, hopefully, there'll be a paper submitted where we use information on genetic relatedness. It's in plant breeding, it's really important because what we do there is we split into additive and non-additive, so we've got to be able to add the two bits back together, whereas mainly with the tree breeding, it's the additive effects that you're getting at. So thank you very much for your time.
Couple minutes for questions.
Thank you for the good presentation. I just wanted to make a comment and then I have a question. The first comment is to emphasize to the group how well our common garden experiments are set up to perform this sort of analysis, because we tend to have a lot of balance, all the varieties or families tested at all the sites, which makes these models is perfect for exploiting these models. The second is with the two varieties that were top performing, you had that crossover interaction depending on those factors. Do you know if the growers could back that into what the actual differences in the environments were? Like, was one a droughty environment associated with some factors? I understand a lot of these things are confounded with each other, but that's oftentimes a goal of determining the G by E, or was it a geographical location?
Yeah. Look, thanks. It's Trevor, isn't it? Yeah. Couple of things. One is, it's lovely you've got balanced data for a start with, but we never have balanced data. Like, it's always incredibly incomplete, but the factor analytic model within a mixed model can cope with that up to a point. Second thing is in terms of trying to understand what's going on with those I classes, essentially, in terms of real environmental covariates. In this particular case, there was they call them hard and soft environments in Australia, so there was some hint at that, but you can spend your lifetime trying to understand what the environmental influences are, and it's lovely to be able to do that. The other way of looking at it, or the way we like to look at it, once again, is in terms of the varieties. I had a variety up there called mace, which everybody's grown for a long time. So a grower will know how that variety performs in their environment. What they're looking for is something that yields higher than mace, but has the same G by E pattern as mace. No matter what happens next year with the environment, they would like to see something, I can't go back, well maybe I can, but that runs in parallel. So it would be lovely to know exactly what's going on with the environments, but if you're confident that a variety is going to do the same, perform the same as mace, and you know how that works in your environment, then that's also the flip side of that, focusing on the variety, yeah.
There's no more questions. I'll hand it over to Nick.
Thanks so much, Trevor. Thank you, Alison. I've got a couple of announcements just before the break. All right, so on your program that you picked up, the 2-page programs there's a little QR code. So just so everyone knows, it's a quick access to the full book of abstracts, which is published online. So if you're sitting there and you want to quickly jump onto somebody's abstract, you can scan that with your phone, and you should be able to pull it up pretty quick. Okay. For those of you that have posters, don't forget to put them up. The poster session will be in this little room called the Okanagan room, just out these doors. And so if you have a poster, it's first come, first served. There's no number assignments. Just put them up and make sure they're up there before the poster session this afternoon. And lastly, after the break, we're going to be breaking into concurrent sessions. So we're splitting the group. Session 2A will be in this room. Session 2B is in the room the third door on your left to the far end of the hallway. Anybody that's talking in session 2A or 2B, please come and bring your presentations and load them up in the proper room before the next session. So during this break, we need you to do that really quickly, please. Thanks. So we have a break now. There's coffee and sweets in the next room, the middle room here. Oh, sorry, just outside in the lobby here. So go ahead, grab something. We'll be back at 10:25 sharp in whichever room you want to be in, please. Thanks.